How to prepare fecalysis stool examination.
Stool analysis, also called as fecalysis or stool examination, is a series of tests done on the feces. Doctors often recommend fecalysis as a part of routine checkup or because they suspect problems in the patient's digestive system. The results can help the doctors diagnose a wider range of conditions from infections, malabsorptions to cancer. In the laboratory, we check for the stool's color, odor, and content such as mucus, blood, microorganisms, or other blood cells. Learning objectives. First, we have the introduction, then we have the advantages of stool examination, collection of stool samples, macroscopic examination, the preparation of slides, concentration of stool samples, microscopic examination, and chemical examination of stool. The normal amount of stools in an adult is 100 to 200 grams per day. Stools are composed of up to 75% water, indigestible residue, undigested food which is digested but not absorbed, bile, epithelial cells, secretion from digestive tract, inorganic material, and bacteria. Take note that our stool is a waste product of our body, so it will have bacteria. Stool analysis determines the various properties of the stool for diagnostic purposes. Usually, we will receive a stool analysis request if the patient is suffering from diarrhea. Now we move on to the advantages of stool examination. Advantages of stool examination. Examination of stools aids in investigation of GIT diseases. GIT means gastrointestinal tract. It is the tract or passageway of our digestive system which leads us from the mouth to the anus. Stool analysis is used to detect parasites. These parasites include worms such as adult worms and tapeworms. In the larvae or ova stage, we also have the protozoa in their trophocyte and cyst stage. Next, collection of stool samples. A random specimen of stool of at least 4 ml or 4 cubic centimeters is collected in a clean, dry container with a tightly fitting lid and transported immediately to the laboratory. So, upon excretion of our stool, it should be transported immediately in the laboratory to maintain the integrity of the sample. For the amount of stool, it should be 20 to 40 grams of formed stool or 5 to 6 tablespoons of watery stool. Parasites are best detected in warm, freshly passed stools, which should be examined as early as possible, preferably within 1 hour of collection. Beyond 1 hour, the stool must be refrigerated. Stool should not be contaminated with urine, water, soil, or menstrual blood. Contaminants may hinder in the result of our fecalysis. Urine and water destroy the trophocytes, while soil will introduce extraneous organisms and also hinder proper examination of stool. Stool sample may be refrigerated if delay in examination, or a fixative may be used if specimen is to be transported to a distant laboratory. So this fixative may include 10% formalin for preservation of eggs, larvae, and cysts, polyvinyl alcohol for preservation of trophocytes and cysts, and for permanent staining. If there's no problem with the stool collection, we are now going to accept, or we are now ready to accept the sample from the patient. The first thing we need to do is to check if the patient already have the payment for the stool analysis test. After that, check if the name in the requisition form of the patient is the same with the labeled name on the stool sample. Then we can log the patient's name in the patient's logbook. We will write the patient's name, the test to be done, and the price of the test to be done. And after that, we can put it in the parasitology section to do the testing. First, we have to do the macroscopic examination. Macroscopic examination of stool includes the stool consistency, color, odor, presence of blood, mucus, and adult worms or segments of tapeworms that can be seen by our naked eye. That's why it's called macroscopic examination.